Okay, so I want to help you fill out your scientific method sheet from today if you missed this morning's Zoom meet. So with the scientific method, the first thing you always have to have is your problem or your question. If you're a scientist and you're trying to solve a problem, then you're probably an engineer. If you are just trying to solve some sort of question of something you don't understand, you're doing something called pure science. Okay? So in order to do any of the steps of the scientific method, you have to have one of the two things happen. So your problem or your question has to be addressed. Step 2 is you gather information, which will help you in deciding how you're going to do your experiment and what you're going to do and what has worked before in the past and what hasn't worked. So one of the things you want to do is you want to look for credible sources, which means you trust them, you believe what they say is true, and they have legitimate data. Not all sources are credible, so credible is super important. The second thing is you want it to be current, especially in science. There's a lot of information out there, but science is dynamic and constantly changing. So if your information that you're using is not current, then your science is not going to be that good. Also, someone else may already be doing that experiment and you're just wasting your time. The third thing is you want it to be relevant. Oops. Okay? And relevant means it goes along with what you are studying and there's a purpose to it and it will add to your understanding of these two things. Okay? Once you've done all your research, and this should actually take a good bit of time because before you do any of these things, this needs to be your biggest part of your pre-lab experience. And then step 3, you're going to write your hypothesis, and this is not a prediction. Okay, when you write a hypothesis, you kind of think you know what is going to happen, but you don't write it that way. You write it if, okay, and then so if I do whatever your independent variable is, then your dependent variable. Okay? So for example, if you are doing a study on plants and you're testing how the amount of water affects the plant growth, so if I change the amount of water, okay, so that is your independent variable because we're going to change the amount of water, then the growth of the plant will change. Okay, notice I am not saying if it'll grow taller, more, there's nothing, there's no prediction. Okay, I'm just saying if I do this, then I will see a change in this. Okay, when you actually do your experiment, you'll be able to figure it out. Now, it's okay that later, before you even start, you have an idea of what you think will happen. So you have a prediction. Most scientists go into their experiment predicting what will occur, but you cannot write your hypothesis that way. And then we're going to come back in just a minute and we're going to talk about if that's the hypothesis, how do you write the null hypothesis? Okay? And we'll get to that in just a second. Okay? So problem, and then you gather information, and then you make your hypothesis, and then you're going to do number 4, which is test your hypothesis. All right, now everybody writes experiment here, but it can actually just be testing your hypothesis. You're testing if you change one thing, will it have an effect on other things? All right, in your hypothesis, you better have a control, and that's your normal situation, and it's what you compare your results to. Okay, and we'll talk about that in just a second. You have your independent variable, and this is what the scientist manipulates. Okay, so whatever it is that you are doing to see what the effect will be. That is your independent variable. Then you have your dependent variable, which is the data you collect. All right, so you know, if you're doing measurements or whatever, all that data that you collect is your dependent variable. So if we're putting it on a graph, we put our independent variable down here, and or time. Time is one of your factors. Typically time goes down there, and then over on your dependent variable goes on your y-axis. Okay, and then with your dependent independent variable, whatever happens, you compare it to your control, and then you have to have constants. Okay, and these are super important. Constants are part of the experiment that you want to keep all of the part of the experiment exactly the same except for your, your one independent variable. Okay, so on the back of this sheet, you have this little experiment, and you have all these little boys, and they all have a word search, and they're all 10 years old, and they're all male, and they're all drinking Kool-Aid, etc., etc., etc. Okay, so those are all of our constants. So 10 years old, male, Kool-Aid. I don't even know how you spell Kool-Aid, okay. Word search, 20 minutes, because it says drink the Kool-Aid, wait 20 minutes, and then time each boy for completing the word search. So we're going to keep every single thing the same except our one independent variable. And that one independent variable is the one thing that's different. And in this lab, is the amount of sugar that they put in the Kool-Aid. So your independent variable is grams of sugar. So everybody gets a cup with liquid in it, but some of them are going to have really, really sweet, sugary Kool-Aid, and some of them are going to have nothing. Okay, so this guy right here, he gets no sugar. So he is considered our control. So boy A with no sugar. Okay, so we're going to say like he gets nothing at all, and then we're going to increasingly give him more and more and more sugar and see what happens. All right, dependent variable, what are we studying? What data are we collecting? The time, because right here it says we're going to time each boy. So time that it takes to complete the word search, okay? And then the problem or question they were probably testing is, does sugar have an effect on your ability to do a word search? Now, there's a lot of other variables that could play. I mean, intelligence and IQ, but let's say they all have the same IQ, okay, so that now intelligence doesn't play into it. Human experiments are really, really hard to do because it's hard to keep everything constant because there's so many effects on humans. All right, so go back to the original page. So now we tested our hypothesis in this situation with an experiment, and then we're going to collect data. All right, when you collect data, all you're doing is putting it in a data table or a chart so that it's all organized. You're not graphing it yet, and you're not analyzing it. So tables and charts are for collecting data. Okay, we do not call them graphs. Okay, our next step is when we do our graph, and then we use these to analyze data. Okay, because a graph should be very easy to read and understand and look at. I'm going to add another part C. Typically, you got line graphs, you have bar graphs, but we also learned about a pie graph. All right, so line graphs will show change over time or constant change. So it could be as temperature increases, as the pH increases. If it's a very sequential measurement, then lots of times we can put that as our line graph. But you want to show a linear change over time. And then your bar graph is for comparisons, right? And so Usually they're averages or estimates or totals, not changing over time. Pi is compare or percentages, okay? And we talked all about how to find those angles and those type of things. All right, and then our last step is our conclusion. In our conclusion, we, you say whether your hypothesis is right or wrong, but the better way for us to do a conclusion now is to accept or reject the null hypothesis. All right, so null hypothesis is important. So let's go back up here. What the heck is a null hypothesis? Null means nothing occurs. So our created hypothesis says that if we change the amount of water, then the plant growth will change. A null hypothesis, the first half stays the same if we change the amount of water, but a null hypothesis states there will be no effect. Okay? No results. Nothing happens. So in this one, your null hypothesis would be if we change the amount of water, then the plant growth will not change. Okay? So when you accept your null, accepting the null means nothing happened. Your variable had no effect. So you have to accept the fact that your variable did not have an effect on your experiment. When you reject the null, you show that there is some sort of something occurring. If the plants did grow at different rates, and we'll learn how to do calculations or whatnot, you reject the null. You say, yes, something happened. I see results or effects. Okay, so we'll go over that as well. All right, so when you do your experiment, you go through all these processes, and they all take different amounts of times. And yes, in your experiment, you're going to have materials and blah, blah, blah. But your main thing is you must have your independent variable, dependent variable, keep everything constant except for your one independent variable, and then have a control where you can compare your results to it. So over here, our regular hypothesis would have been if we change the amount of sugar, then the rate at which the boys do the word search will be different. The null hypothesis would say if we change the amount of sugar, the rate at which they do their word search will not be any different. Basically, we would say the sugar has no effect on the word search. Okay, so null equals no effect. Okay? All right, so I'm going to stop that right there. I think you know here we have our dry mix, dependent, responding y-axis, manipulated, independent x-axis, and that's what goes here. Sometimes people graph the control so you can compare, and then sometimes people don't graph the control. So we'll talk about what you would do about the control on a graph later.